So we were at the stage, should I start? Yeah, yes? Okay. So we were at a stage yesterday where we were going to discuss the, balancing the kinds of broad defense that you might get from having a larger repertoire against the problem of autoimmunity. And what had we written down? We had said that you have this spacer system and you have some self-targeting probability, which is equal to some combinatorial factor alpha k fix k var, where k fix is the number of mutations, number of mismatches you'll tolerate at fixed positions. K var is the number of mismatches you're willing to tolerate at variable positions. And L is the length of the, the whole immune element. So spacer plus PAM. Yes? So that's what we said. And then you get to multiply this times L, because there are many places where you could match. L is the length of the bacterial genome. So there are many places that you could match. And then you get to multiply by 4 to the minus L, because a perfect match requires, you get to pick 1 out of 4 nucleotides. Okay, that's what you said. And then we also, I think, worked out what this alpha was supposed to be. Remind me if I'm incorrect, but we said, I think we worked this out. I think we worked out that the combinatorial factor, yes, here we go. We worked out that the combinatorial factor is given by 4 to the power of k fix and L minus k fix, k var, and 3 to the power of k var. Okay? And then we took some logs and stuff like that. Now, also recall, do I need to put that down? We'll see if we need to put it down. Okay, so this is, we also said that this thing here is approximately equal to L minus k fix to the power of k bar. This is all we need. So we also said that this is all derived under some assumptions, of course, of uniformity. Everything is used equally often. There are no correlations. And of course, you can also correct all these things to include those effects should they be considered to be, if you find that they're important. So now what I want to do is, this is the probability of autoimmunity, of getting an autoimmune problem. And then there's, of course, if you've got more spacers, you have a better chance of beating off the viruses. So we want to balance this in the long term. So we're going to do this as follows. So there's a model I'm going to write down for deriving a balance between immune defense versus autoimmunity. Okay, so let's go after this. So the idea is, let me set up the model. As usual, I'll state my conditions and what I'm going to do. Then I'm going to write down some equations. Then I'll do something. So we're going to consider a microbial colony under episodic attack. So every once in a while you have a viral attack with probability Q. So we're going to imagine some sort of discrete time setting where generation by generation by generation, each generation, your probability of getting a viral attack is Q, that kind of thing. Then the idea is that many individuals die, but some survive, and they grow. So from the population point of view, you have and then lineages with a specific spacer for the threat are more likely to survive. Okay. So then we also realize that the probability of a defense equals the probability of defense increases with n equals the number of spacers. At least up to a bound set by the number of casts. Remember, we said in the previous lecture we derived this sort of optimal number. You could have, you know, except that the optimal number seems to vary quite a bit, right? You know, between a few tens to a few hundreds. So now we're trying to understand a little bit more the details about that a few tens to a few hundreds. So we'll allow some of them maybe have more casts, some of them have fewer casts, etc. And as a result, there's you could have many numbers of spacers, and you know, you could by the way, you could increase what's optimal from the point of view of yesterday's lecture by just increasing the number of casts. If you double the number of cast proteins, well, it would be optimal to have twice as many, right? From that point of view. So then what fixes the number of casts then, if you could ask. So anyway, we're going to try to work out this balance like this. Yeah? Okay. So the n dependence of P-def, of the probability of defense, could be complicated. And it could be complicated for many reasons, because there is host phage coevolution, which you already mentioned. And for example, and other factors. You can imagine this to be a complicated function in general. But one thing I'm pretty sure of is that it should be sublinear, in the sense that you should get diminishing returns from increasing n. Now, why is this? I mean, you can say many reasons. Just because I have a second spacer, it's not going to be doubling my chance of having a defense, because if two spacers are attacking, for example, the same virus, you know, we already know that having a second spacer doesn't necessarily help as much because the first one is already working, right? Or it may be that the first spacer, while mostly bound to this virus, binds a little bit to that virus, because there's, you know, always some cross-reactivity in these things, and therefore it's already giving you some defense, so it's not really going to double the chance that you survive. So in general, these things are always sublinear. There's a law of diminishing returns usually. You know, you put double the resources and you get less than double the return. It may help, but it's not going to be double as much. So given our ignorance, so the standard trick as a theorist, if you're ignorant about something, is to parameterize your ignorance. So that phone is, again, going beep, 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 because yesterday, everybody had midterms, you know, the kids and everything, and today is my wife's birthday, so everybody in the family is sending messages saying, happy birthday. That thing is beeping. Okay, so I'm going to parameterize our ignorance, so that's the standard way of going about things, and I want my parameterization to kind of reflect this law of diminishing returns. So if you think about it, the way, you know, we're going about all the theory that we're doing is we're not actually trying to model every detail. We're trying to model the parts of the problem that we think are going to be significant, right? So that's what we're doing. And then, of course, you may turn out to be wrong, and then you have to go back and revise the model and everything, but that's just the nature of life, right? So I'm going to say that this is equal to n over beta, some number, and I'm going to raise to the power of gamma, and beta comma gamma are fixed parameters, and I'm going to imagine that in general gamma is less than 1, so that this is p def as a function of n will, you know, do this kind of thing. Okay, so that's just trying to capture somehow the law of diminishing returns very roughly. Okay, so I can also say that increased protection of this kind is balanced by autoimmune risk. So let's write an expression for that. So the idea is that, well, the probability of, if you like, how should I write this? I'll write this as auto survival. The probability that you survive your autoimmune risk is going to be equal to 1 minus P self. So remember, P self is the probability of self-targeting by one spacer. So 1 minus that is the probability you survive. I'm going to put an E here for efficiency. And that's because just because the spacer can target you doesn't mean it'll succeed. Remember, we said that even when you're attacking viruses, there's some statistics to whether or not the CRISPR machinery succeeds. So I'm going to parameterize that also as some parameter E, which is going to be less than 1. And then I'm going to raise this to the power n, because you need to, it has to be that every single spacer, spares you. So I'm going to add them all. So I'm going to put them all in. So we get this. And right. So naturally, I've made many assumptions here. I've assumed all spacers have the same length, same P self, etc. Whereas actually, it's the case that, as we'll see in a little bit, the different CRISPR types have different properties in this regard. So there are some CRISPR types which, like CRISPR type 1E and 1F, they have very narrow spacer distributions. It's all around, I forget, 32 or something like this. So this is all perfectly correct for that. But CRISPR type 3 has a very wide distribution, kind of peaks up near like 28, 29, but actually it stretches all the way down to 22 and stretches up to 35, and there's a long distribution. Some of them are long-tailed. So, so a refinement that you could definitely do here is you could fold in those kinds of distributions. I won't, and then I'm going to argue to you at the end that I'll give you some evidence from the data that in fact, all of that works out correctly, that in fact those CRISPR types that have larger mean spacer lengths are indeed suppressed more or included more in certain cases. I'll break this down with more nuance towards the end of this section. Right? So as you can see, our method is we take very simple approximations, we get some answer, then we refine it, then we refine it in that way as a systematic way of including the kind of biological details that matter to the individual kinds of species. But for now, we're going to take this and move along. So now I want to build my model of how to derive the optimal repertoire size. That's related to the question from yesterday, except now instead of just a single number, oh yeah, it should be this range, I want to sort of go about it with a little bit more nuance, what would set for some sort of individual species, perhaps the operating point, right? So that kind of thing. So I'm going to go after this. And I'm going to do this as follows. I have this probability of defense, and I have the probability of survival. And so I'm going to use these to model the long-term population growth in this colony by noting that, with the probability Q, you encounter a virus. And when you encounter a virus, having this probability of defense in the game will help you. And at all times, whether you encounter a virus or not, there's some chance of autoimmunity. So we can just sort of work out, you grow well in periods where there's no virus. You do sort of badly in times when there's a virus. Then you grow again, then you go down, and there's some bumpy pattern. And in the long run, there's some growth rate that the system should achieve. And we'll find out what that growth rate is, and we'll try to maximize that. Okay. So we're going to basically take a discrete time model. And I'm going to say that the expected number of offspring per generation, well, let's see. I'm going to call this FT. And this is at, if you like, time t. So this is generation t. And t is, 1, 2, 3, like that. So that's going to be equal to, let's see, e to the r max. This is the maximum number. max value of the number of offspring you could get, right? I'm going to parameterize it this way, right? So this is nobody's dying and everybody's reproducing, right? That kind of thing. And then I'm going to put down here a probability of defense with an xt here. And xt is a random variable which takes values 1 if you have an epidemic, and it takes the value 0 if there's no epidemic. So that's because, so on the time step t, if there is an epidemic, well, the probability of defense should appear here in your probability, to tell you how much the growth is expected to be. And if there's no epidemic, it's not there, right? This is just a factor 1. And then I have to also multiply here by the probability of survival of autoimmunity. So that's what I expect to be happening in each time step. Okay. So we also said that xt equals 1 with probability q. Okay? So basically, this is a model that's describing a succession of booms and busts in the population. Okay, so the population size at time t is then given by nt equals the product, let me put down n naught, which is the population at time 0, what you start with, times the product from I equals 1 to t of ft. Okay? So this is a model that is sort of valid kind of in the large population setting. Because if there were small populations, then we should also worry about sort of fluctuations, stochastic fluctuations, which could lead to, for example, extinctions. But here I'm taking this it's all continuous. You're allowed half a bacterium, stuff like that here. So it's all very continuous. If this came down to small numbers, then you have to start worrying about actually including the stochastic fluctuations with greater care to allow for extinctions. But here there's large populations, so we can just let this run. Okay, let's work out the long-term growth rate. And that is, the long-term growth rate is going to be given by lambda equals the limit as t goes to infinity of, I can take the logarithm of nt over n naught, because the long-term growth, if you left the thing along, is exponential, right? You grow by a factor every generation. So I just want the exponent, the effective exponent that I'm going to get in the long-term growth. And here I haven't capped the population by imagining that I have some carrying capacity in my experiment or something like that. So it's log nt over n naught. And then I get to divide by 1 over t, because you're expecting e to the alpha t or e to the lambda t in the long run. So I'm dividing out the t. Excellent. So lambda is a pretty good measure of the success of the strategy that you're using. The strategy here is understood to be, your organization of your CRISPR repertoire. So it's that strategy that you're playing. So if I look at this, I see as nt is equal to n naught. Well, we already wrote down what that is. So we have that formula there for nt. Where is it? We have the formula there. We see that lambda is equal to the limit as t goes to infinity of 1 over t times the logarithm. I can remove this. The sum on t equals 1 to t of the logarithm of ft, because, of course, the logarithm of a product is the sum of the logarithms of the terms. So now we apply the law of large numbers. And the law of large numbers in this case says that lambda is going to be equal to 1 over t times the sum is in the large number, in the large sum limit is going to be equal to t times the expected value of the log of f of t. Right? So great, the t's cancel. And I want to work out this expected value. But hey, that's really easy. Because there's a probability q that there's an epidemic. So that means if there is an epidemic, I need to take the example with xt equals 1. That determines the growth rate. Or there may be no epidemic with a probability 1 minus q, in which case I should put log of f xt equals 0. Easy. Now I remember that f of xt equals 1, looking at what we had above, is going to be equal to e to the r max n over beta, to the power gamma. Here I'm using my rough parameterized assumption about the benefit you get as satisfying this law of diminishing returns. Really, what I really care about here is it sort of goes like this, right? I don't really care about exactly how or what. That's tuned by beta and gamma. And I have a 1 minus e p self to the power n. And f of xt equals 0 is equal to e to the r max times p auto survival, which is going to be equal to 1 minus e p self to the n. Okay, let's put this together. So we get lambda equals, okay, I'm going to write out the terms. So how should I do this? Okay, so here is the way to do this. So if I take the log of this, one term I get is r max. Log of this, one term is r max. This r max is multiplied by q. This r max is multiplied by 1 minus q, right? So q plus 1 minus q is 1. So you get a term that looks like r max. So that's the first term. Let's work out the second term. So with the probability q, this term is there. If I take the log of this term, that's gamma log n over beta. So I should get a plus q, q gamma log n over beta. And plus, then I have, in both cases, whether I have or don't have an epidemic, I have this term. So that means I get q times the log of this plus 1 minus q times the log of the same thing. So the q plus 1 minus q adds to 1, and I just get the log of this. And the log of this is, by definition, n times the log of 1 minus e p self. Nearly there. So this is approximately equal to r max plus q gamma over log n over beta. I just want to make one more simplification here. Plus, well, if I look at this, this is a small number. The probability of self-targeting, although we said it was large enough to be significant, it isn't vast, right? I mean, even at the best, after really, we got it to about 10 to the minus 3, 10 to the minus 4. So 1 minus a number of that, and then there's this e. So this 1 minus a small number like that, if you take the log, is approximately that number. So you get minus n e p self by approximating the log. So that's our expression. Our goal is to optimize this. And we'll do that in a second. Okay, so we're nearly there. I'm doing this to make you happy. We're going to derive this all the way to the end. Okay, so let's take d lambda dn equals 0, because this is the maximize the growth rate. So the idea here is that the parameters of the CRISPR system will get selected over time in order to maximize the growth rate, which we're taking as a figure of merit for how well the CRISPR is organized, right? So I take d lambda dn equals 0. And what does that give me? If I look at this, you know, there's only one n here, and there's one n here. So differentiating that, I get q gamma over n. The first term differentiates to 0. The log here gives me the 1 over n. And I get the q gamma over n. And then this thing differentiates to give me a minus n EP self. I'll move it to the other side of the equation. So I'm going to get EP self. So that's our condition for maximizing the growth rate. Okay. So now I'm going to remember what P self was. So this was going to be, oops, there's something off here. Oh no, this is fine. I want to sort of reorganize this a bit if you don't mind. I want to write this as log n. I'm going to move the n to that side and take the log is equal to log of q gamma over EP self. And the reason I'm doing this is, remember, P self has various exponentials in it. So I want to drop the exponents. So that's why I'm doing that. And so this is equal to the logarithm of q gamma over, well, in the denominator, this is E. And the probability of self-targeting involved the length of the genome. It involved a 4 to the minus L, you know, because it's how many ways there are of L being the spacer length. And then there was this combinatorial factor k fix, k var, comma L. Okay, so now let me work that out. So I have here, okay, let me first write this out like this. This is going to give me from this term, it's going to give me an L log 4. That's this term, taking the log of that. Then I'm going to get a minus log of alpha of k fix, k var, and L. And then I'm going to get a constant, which looks like minus log. And I'm going to put the EL in the numerator. That's how I got the minus sign. I'm going to flip it, the argument, over q gamma. Easy enough. OK, the remaining work to do is to compute that. So recall, did we write down alpha here somewhere? No, I need to rewrite it. So let's recall that alpha of these three arguments is approximately equal to 4 to the k fix times L minus, this is the spacer length minus k var, the number of variable mismatches, to the power k var times 3 to the k var. Okay. Remember, k var is the number of variable mismatches, k fix the number of fixed mismatches. So using this and linearizing the result around characteristic spacer lengths, like about 30. And the reason I'm doing this is the actual lengths of spacers don't vary very much around that, right? They're like 20-something to 30-something, like 40, occasionally you get something longer. So it isn't like they vary over orders of magnitude. If they varied over orders of magnitude, you need to keep the full nonlinear expression. But around some range, you can kind of linearize, and so that'll reveal what's going on easier. So I'm going to linearize it. So all I've done is I've plugged this in, taken the log, and linearized. Okay? So you plug it in, take the log, and linearize, and you get the following answer. Log n goes like L times log 4 minus k var over L naught minus k fix. That's it. And so if you pick, let's say, k fix, L naught, like 35, like typical numbers you find, that's like the typical length of a spacer plus PAM. You know, PAM is like 3 things, and the spacer is like 25, 26, 27. You know, add it up, maybe 30, you know, something like that. It'll be around 35. And suppose you pick k fix to be like 5 and K var to be like 5. There was a discussion yesterday in which I told you why I'm picking these numbers. Each CRISPR has different K fix and K var and details. We decided we're not going to worry about the details of each one separately. Roughly, it's like 5 each, roughly. We're trying to get to the ballpark, and then details we'll worry about by CRISPR type another time. So if you plug all these numbers in, you're going to get a prediction. This is 1.2L. So this is the main prediction, right? But this is a further prediction that says everything should be sort of lying around numbers like close to 1. Maybe we shouldn't care about the first decimal place. Maybe we should say it's close to 1, right? Something like this. A little bit more than 1. So another way of saying this predicts, we've just predicted a scaling law. And the scaling law says that n, which is the repertoire size, ought to scale like e to the 1.2L, which, I'm going to write more evocatively as 3.5 to the power L. The reason I'm saying 3.5 to the power L is, by the way, if you did the completely naive thing and pretended that even without the mismatches, the autoimmunity was a very important thing to take care of, then you can do many hand-waving, easy, 3-line arguments that suggest that you should have a scaling that goes like 4 to the L, right? But that's corrected because you really have to care about the mismatches and everything, and the scaling is more like about 3.5 to the L. That's a very nice thing, right? Scaling laws are not easy to come by, and this is a predicted relationship. And if you predict that, you should be able to check it in the data because there's a lot of sequences. So this is where a biologist would say, I should be having slides and projecting it and show you the real data, but I'm going to draw the data, and it's just as good because I could fake my slide too. So it's just as good. So I'm going to draw a picture of the data. You can look at our paper, which I gave you the reference for the actual data and everything like that. And referring to the case where you asked for code and everything and wasn't given to you, in this case, we've posted our code publicly. You can get the data and the code. It's all packaged up.
And the language was an open source one? Was a free one? Because the one...
Python. Which all the cool kids use.
The one I was requiring even was in MATLAB.
Yeah. No, MATLAB is a complete headache. So, many experimental labs use MATLAB. And I've been slowly, I give a class that's in MATLAB because the students have it for free. Okay. But, the grown-ups don't. Yeah. So I've been everybody's slowly migrating. And basically, the students in my lab don't want to use anything but Python because who wants to pay? Yeah, so anyway, so there's a migration away, I think, but we'll see. Now, MATLAB is going to, if they see this video, they're going to get mad at me. Anyway, so it's 31 to 37, okay? And this is log scale 10, 100, 1,000. And you get a picture that looks a little bit like this. It's definitely not perfect or anything. And there's a little bit of, I'm drawing binned data here. But you get a line going through. This is, so plus is binned data. So I'm drawing the binned data here because there are about 2,577 strains in the last edition of the CRISPR database. So there's a database that you can go to if you're interested in playing with these things. There's a database where all the known sequences are deposited, and it's updated every once in a while. The iteration we used had about this many strains that have been sequenced. So you look at the bit, so this is binned for visualization, but we're going to fit to unbinned data. And by the way, I want to emphasize, I'm not really talking about data analysis in this talk, but I want to emphasize that if you do data analysis like this to fit things like scaling laws, it's actually important that you fit to the unbinned data. For visualization, because there are lots of points, it may be very useful to bin your data, but the unbinned data is very important because if you play with it, you'll rapidly see that by binning the data in sort of peculiar ways, you can produce I have to take this call. I'm sorry. Hey, So, all right, great. So there's a fit to the unbin data. And here is the icing on the cake. It turns out to give you 1.1 plus or minus 0.1. Ta-da! Times L plus constant. So great, right? There's only been a couple of times that I've had the experience where you make up a theory completely ignoring the data, and you do the thing, and then you match it, and it kind of works. There was another one I did on grid cells, which I didn't have time to tell you. I just love this kind of stuff when it actually works, right? So actually, sometimes I tell students, really theory students, like I had one math student who worked with me to talk about the grid cells in the brain. This is a paper I'm very proud of, which appeared in eLife about the organization of the spatial map that you build in the brain. And we found interesting scaling, and there was a square root of E that appeared somewhere that I liked because I liked the number. We did this, the calculation took like 5 minutes or 10 minutes. And then the student said, well, we can compare with the data. And this happened twice actually in the grid cell thing. And every time a theorist says that, I say, well, if you actually compare with the data, it'll be thumbs up or thumbs down. And often it'll be like this, right? And then you have to decide before you do the analysis what you will do in each of these three cases, right? If it all works, you're done. Great, you have a hit paper. If it's no, then it doesn't work, but you may be able to publish the work anyway. I'm sort of giving advice to theorists here. You may be able to publish the work anyway as an interesting theoretical exercise, because there's a rule that often applies, especially in theoretical physics, but certainly in theory in general, that anything interesting, if it's a non-trivial result, will eventually find its place. It may turn out to be wrong for the thing you were modeling, but eventually there'll be something for which it's correct. So it's actually important to find a venue to publish it, because someone will find a use for it. So that's an advice I give to students, because otherwise they often get very depressed if something doesn't match. But very often when you match with data, you're kind of in this sort of intermediate kind of setting. And then you have to think very carefully about how you pitch it, how you explain why it may not be matching. People like to see stuff that matches, but things that doesn't match are good too, because they teach you about the system. So then it's subtle writing, and the art of writing, scientific writing, becomes really important in communicating that work. But this was a case where the thing matched. So this is good. So then you can do cross checks. So for example, there are many species that don't have Cas, but they have CRISPR, right? So in other words, there's a CRISPR cassette, but there's no way for them to use it in terms of memory. So presumably in those cases, there should be no association between spacer length and repertoire size. So indeed, you can check that in this database too. So you have log n, and you have spacer length. So I'll put this as L.
Professor.
Yeah, hold on one second. In this case, you find something that's sort of more like this, slight negative decline. If anything, it's sort of down this way, but there isn't much of an association. So that's good. There are fewer strains like this. So there are about 344 strains without Cas. So that's great. So that checks out too. Okay, so I'll take the question in just a moment. The next thing that I want to start putting up here before I take the question is the question of mechanism. So that's a win, right? We built a very conceptual theory involving the statistics of immunity and stuff like that. It predicted a thing on very, very general grounds. The thing not only is there if this coefficient, this exponent hadn't matched, but there was a scaling, I wouldn't have cried too much, because there are many, many assumptions that went into the actual number. It's the scaling that is the real prediction, that there is a scaling. And there is a scaling for sure, right? But what do you know, the number matched too. So it's icing on the cake, and it's kind of nice. So great. So once something like that works, then you really want to dig into the details. Something interesting is going on. You want to understand the biological mechanisms. How does the scaling get enforced? How is it selected for? Actually, as we said, things differ between different CRISPR types. So now we should, we should look at the different CRISPR types and see what goes on. So I'm going to give you little vignettes from the data about that. We won't go into detail because I want to switch into talking about another topic. But there was a question from somebody. Did this calculation meet your approval?
Yes, I'm very
Yeah.
truly happy today. It's amazing how it fits the data. It's
good,
really
good.
nice.
I'll tell you another one in the break that's really cool.
Nice. My question is about these organisms that don't have Cas. What do they use CRISPR for?
They don't appear to be really using CRISPR, or they may be, but we don't know. So really, so there are two things you could say there. So, you know, there are various kinds of anti-CRISPR mechanisms. So it's possible that the kind of thing that happened was they acquired some autoimmunity out of it, and then so very rapidly that it was selected out. So you got another thing you could check is if this happens, is there there's a higher incidence of self-targeting spacers in it. And so we did a statistical analysis of that, which is a little bit harder to do, and there was indeed a higher incidence of self-targeting spacers. So it's possible that the mechanisms for turning off CRISPR or Cas, right? You know, to turn off CRISPR, you can turn off CRISPR, you can turn off Cas, it doesn't really matter. If you turn off Cas, there's no targeting. So then they're hobbling along using the innate immunity mechanism. There are other mechanisms of immunity too, which I'm not really analyzing here. So one interesting open question is to expand this model to include the other kinds of innate immunity that bacteria have, right? Including restriction enzymes. You know, they've got inflammatory responses, various ways of helping their surfaces to not allow the viruses in, stuff like this. And the reason why I think it's very important and interesting to include that, besides for the study of bacteria, is really I actually first started working on vertebrate immunity, and then switched to working on this, first of all, because it's super cool, and secondly, it's very clearly defined. Vertebrate immunity is just much more complicated. Many, many systems, T cells, B cells, various inflammatory systems, cytokines, you know, blah, 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 blah. You know, you can regulate this in a dozen different ways. You produce the antibodies, but then, oh, wait, if it actually matches a threat, then you expand that, you know, there's this expansion of that, then you create a memory compartment, you know, there's just lots and lots of phenomena. So what actually happened for me was I realized, wow, there are many, many, many dynamical phenomena. I just need to work on a simpler system to get my head organized about what kinds of things I should be paying attention to, and then switch back. So clearly, the next things to worry about here are things like the coevolution and these other components, and how all of them interact together collectively to achieve the effect of immunity. Because getting the techniques for doing that, once I have a little bit more, well, any of us has a little bit more experience with that, one could go back and study the even harder case of vertebrate immunity with some experience on how to deal with coevolution, multiple systems interacting at the same time, and so on. Does that answer your question? Yeah. Very good. So what is the mechanism? So I'll talk about two kinds of things. So first I'm going to draw a picture that's kind of very interesting. So here is CRISPR type 2A. And it turns out that, so what's this? This is around 25. And this is sort of a spacer length. And this is kind of like around 50. So CRISPR type 2A is kind of very narrowly focused down here. This is 2A. CRISPR type 1E, it's kind of very narrowly focused, a little bit bigger, around, let's say, 25, 27 or so. I should make this even narrower. 25 to 27, that kind of thing. Then there are many, many types. You know, we enumerated already 3, 6, 9, 12 types of different CRISPR. There's even more floating around, but these are the ones for which we got data. So then you have type 3B, and here's 25, and here's 50, and this sort of does this and goes down like that, very, very wide, right? There's another one here, 3A, which is looking like this, like that. Now, interestingly, by the way, type 3 CRISPR doesn't use a PAM. And so there's something going on involving the uses of type 3 CRISPR, which aren't exactly the same as the use of type 1 and type 2. And there have been recent papers suggesting that type 3 CRISPR actually plays different roles. So it may be being used for other things. You know how everything in biology is borrowed for multiple stuff. You know, any given pathway does 6 things, right? So it's possible that this is doing 6 things, you know, for all we know. And that's why it's organized as we don't know, right? Anyway, so there's these differences. So you could ask, given these differences, you might imagine some of these things have smaller spacers, And some of these things have larger spacers. So you might imagine that if you want to use CRISPR type 1E or type 2A, then you need to have a smaller repertoire because the spacers are smaller. So if it's true that this association, like we're talking about, is true, then there ought to be an association between some, CRISPR types with smaller spacers and smaller repertoires. Hey, that you can check too, right? So this is the great thing about having the data. You can sort of, even if you haven't built a model fully, you can quickly go check these things and see it doesn't make any sense. So I'll draw a picture of that. This is really nice. So I'm going from 20 to what? Maybe about 60 or so here. And then, so this is repertoire size. This is like N. And this is like, I don't know, from whatever it was, 42 to 32. There's some numbers here. And so I'm drawing the mean sizes and everything. So there's a 2A. Anyway, I'm not going to put out all the details. It's not a perfect association, but there's a line going through. Exactly the kind of thing you'd want. Now, so we haven't attempted to sort of predict in detail what these things should be, because basically this was already a long paper. That's the first reason. But the other reason is that you need to know details, right, about many things here, about the PAM. So there's lots of specific details about each type, which we didn't really have ready access to. But that's an open area to think about. Another thing you can do is, so that's A. So there's clearly CRISPR type use. Selection of CRISPR type use is involved in this process. And B, you can also write down dynamical models, dynamical birth-death models, and show that if you can select on, for example, the CRISPR acquisition rate in these birth-death models, you can, in fact, generate scaling laws like this pretty easily. So there are ways of doing this. I'm just saying that, to get this, you need to have a selection mechanism, and selection needs a knob, a thing it can act on. You can't just act on nothing. There has to be a thing it can act on to produce the scaling. So I'm just saying that it can act on, for example, selection of CRISPR subtypes, right? It can also act on, for example, dynamics of acquisition. And no doubt it can act on stuff like details of the biochemistry and this, that, and the other. So there are various ways in which indeed you could select for this. This is just a proof of principle that there's something to select on. Great. I'm going to stop here with the adaptive immunity. Next, I was going to talk about vertebrate immunity, but I think I feel sort of morally bound to say something about neuroscience, since that's supposed to be my main area. So we're going to switch into talking about neuroscience. Any questions about this material? Okay? All right, so then I'm going to move over there. And while I'm erasing, I should say that you're actually going to get a number of lectures about theoretical and computational neuroscience from various people, including Professor Mauro Copelli. And so because of this, I'm not going to give my standard introduction to the subject. So my standard introduction to that subject would involve starting with telling you what a neuron is, talking about the fact that neurons are electrical circuit elements. We would discuss the famous Hodgkin-Huxley equation, which won a Nobel Prize for modeling neurons. Then I would say, well, you can also simplify the Hodgkin-Huxley equation, and there are very simpler models, because that's so complicated. There are very simpler models, such as, for example, there's this beautiful thing called the Izakovich model made up by this guy named Eugene Izakovich. He has a great book on the dynamical systems modeling of neurons. It has like 5 parameters, and it will capture the dynamics of essentially every neural type. So that's what, in my lab, we no longer even look at Hodgkin-Huxley type modeling. We mostly just use Izakovich neurons and then fit the parameters in the thing, and it'll reproduce the right dynamics. So it's an effective description in the sense that I talked in the first lecture, right? There's the detailed model, but actually to capture the structure, the dynamics, you kind of need 5 parameters, right? So it's a beautiful thing. So I would advise to look there. And then what I would do is, okay, great, let's put this thing into networks, discuss dynamics of networks, etc. Professor Copelli will talk a lot about that. So what I'm going to do is instead I'm going to take the kind of approach I've taken with CRISPR here to discuss certain aspects of the sense of smell. And you'll see the ways in which there are analogies to be made between the immune system and the olfactory system. That's why I'm picking the olfactory system as a thing to segue into from the immune system. So let's talk about olfaction, which is equally the sense of smell. Okay, let's see. All right? So remember, the CRISPR immune system worked by matching gene sequences. So that's a very specific, easy to think about structured things in A, T, C, G, whatever, that's kind of easy to imagine. But the immune system of both vertebrates and the olfactory system are not matching sequences like that. So vertebrate immune systems, and animal olfactory systems, it doesn't have to be vertebrate, olfactory systems work by detecting three-dimensional molecular shapes not linear gene sequences. So this is a much, much harder problem, right? Because it's some complicated thing that floats around, makes some shape and stuff like this. So if you think about it this way, really the sense of smell is the sense of touch. What you're trying to do is feel the shape of a molecule. You're just trying to do touch at a molecular scale. And, okay. So, well, if I draw a cartoon of what this is supposed to be, well, here's one shape, and here's another shape. So these are epitopes, as they're called. An epitope is a piece of a molecule that you bind to. So the full molecule is whatever it is. But then there's this little piece that you may be seeking to bind to. And then you may have one receptor, which has a sort of pocket like that. And then you may have another receptor that has a pocket like that. Right? And of course, this is a cartoon in like 50 different ways. So for example, both molecules fit both pockets, sort of, by which I mean really you think about the binding affinity between the molecule and the receptor, and basically everything binds to everything at some level. It's just that some things bind well and some things bind less well. So you're going to get all kinds of cross-reactive phenomena where the same receptor binds to many things, and you've got to worry about their binding affinities, and so on. Worse, or more complicatedly, or better, you can have multiple binding sites. So the same receptor may be able to bind to multiple things at the same time. Right? So you could have that. Then you can have phenomena like cooperativity, where you have one molecule which binds to a piece of the receptor, but doesn't really trigger a response. But the fact that it bound to a piece of the molecule means the other thing binds, right? Because it changes the shape of the pocket, right? There's sort of tons of phenomena of this kind of headachy, right? Now, it is the case that you do get molecules and receptors that have been selected over evolutionary time for precise, specific binding. So, for example, this is the case, for example, with hormones, which are sort of such important switches in the body that it's really important that you don't screw that up, right? And so then it'll get selected over time. Even other things, like, for example, and this happens also, for example, in smell, maternal bonding, smells, right, are processed separately from other kinds of smells. So there's a certain class of smells which are very important for your innate behavior. Stuff like you smell this, you run in fear. You smell this, you feel affectionate towards with the pup, whatever, for stuff like that. So there's a separate pathway going through a region called the vomeronasal organ where some of these things are processed. It's not just us, it's even flies. Flies have a separate pathway for these kind of innate behaviors with strong, specific binding. But most of the stuff you want to do is kind of more general. I don't know, it's not that important that I smell the apple precisely, right? It sort of just binds to lots of stuff. So in general, there are examples of strong specific binding, but in general, kind of everything binds to everything. And so you already know before you start that whatever you're going to do with olfaction, the character of the problems you need to solve is going to be sort of different from what we've been talking about in CRISPR. So in CRISPR, the problems had to do with there's this very diverse space of viruses. And you need to store things that are specific to these different ones, and you're trying to, and there's dynamics, virus comes in, virus goes out, it can kill you, things like this, those kinds of things. Your problem here is going to be, well, you have a very large space of molecules, we'll talk about that. You're going to have a certain number of receptor types. It's not going to be as big as the space of molecules. Somehow, everything will kind of bind to everything, so you've got this horrible multiplexed kind of signal, and you've got to figure out from this what the odor was. So it's going to be combinatorial coding is more like the problem you need to solve. Okay? And the point of the 1.5 lectures left to go is going to be the following. So it turns out that, so I would say that the olfactory system is a formal circuit for solving this problem of combinatorial coding of odors. So that's what it's there for. So sort of astonishingly, it's actually mind-boggling. I will draw for you later the circuit pathway that's used to do this. And basically, in Drosophila and human and elephant, right, it's basically the same. So the biochemistry of the components is different. Some details about how the circuits are built and everything is different. But as you'll see, the computational logic and organization of the circuit is identical. Which tells you, and these are such, flies and humans are such separate lineages, we diverged so long ago, that tells you that there is something about this circuit architecture that's powerful in processing these kinds of signals. So given that conservation of the circuit across the lineages, we're going to go after the question of the information processing and computations involved and why the circuits are organized the way they are. So that's what we're going to go after. And we'll use at least some of the similar techniques from the previous part of the lecture. Okay. So if we want to think about this, we need to think from the front end all the way deep into the brain, right? And the front end, before the front end, is the actual space you're trying to sense. Remember, before we started talking about the CRISPR mechanism, I said some words about viruses. I couldn't say that much about viruses, but we said something about their diversity, this, that, and the other, right? And then many sequences and so on. So let's start by talking about the structure of odor space. And so the first question to ask is how many odors are there? Right? So normally an odor is a mixture, but we should start with single molecules. How many molecules can float in the air? So there's an answer to this, and there are maybe, I don't know, 10 to the 4th to 10 to the 6th volatile molecules. And the reason I'm giving you such a big range is because different authors give you different ranges. Because it depends a little bit what you mean. If the stuff sinks near the ground, bounces around, maybe it's relevant to dogs that sniff down. It's a little bit iffy exactly what you want to mean. But certainly there are at least 10,000 different molecules, right? So when you think about these molecules, how are we getting so many molecules? All the little ones don't matter, right? I mean, it's like little bits of with 2 atoms. We're not talking about that. There are not that many of those after all, right? We're really talking about stuff with. The way you get lots of molecules is things with lots of carbon chains and everything. Every aldehyde has something at one end, and it's got a long carbon chain with various kinds of side groups. Every ester, there are lots of smells that are esters, have something at one end and something here and something there, and then a long carbon chain. And then what happens is that all of these things float, and indeed animals and plants use esters and aldehydes and ketones, and that's how they make these kinds of different kinds of smells. They make these long things and they attach something here and something there to give it some shape, right? And then that's what they give you. And if you make the carbon chain too long, it's too heavy and it won't float. So there's some bound to how big the molecule can be. That is why this isn't an unbounded number, right? But anyway, for our purposes, it's just big, right? Okay, it's like 10 to 10. So these are the number of things you want to sense. However, it turns out, or maybe in addition, natural odors, let's say foods and fragrances, have about 40 important components. So how is this known? So, you know, the artificial flavor industry and scent industry is very, very interested in this because they, of course, want to make artificial flavors that will taste to you like strawberry or smell like something they can put in Chanel No. 5, you know, and get it cheaper than getting it from the plant. So to do that, they have to know what the molecules are that give you your percept of the smell. So they have done extensive catalogs. So they'll go through these smells. They'll do mass spectroscopy to figure out which molecules are there. Then they'll produce mixtures of the stuff and try to tell people, does it smell like strawberry? You know, this kind of stuff, right? And it's like 40 to 50, let's say, things that are mostly important. You know, there's slight details beyond that. There's also a very nice paper from the group of Noam Sobel, who's a professor at the Weizmann Institute, in which he did a bunch of experiments where he said mixed odorants and asked people to say, are these two different? Are these two different? This kind of thing. You know, you can't really very easily say what's in an odor. If I tell you what are the components of this odor, it's kind of hard unless you're really trained. But you can ask if things are different. You can sense things are different without knowing what's in them. And so it turns out that around 40, 50, this kind of thing, odors, he named the phenomenon olfactory white. They all start smelling kind of, let's call it white for lack of a better word. Right? You know, they call this kind of smell the same, right? It, as a thing. Actually, my daughter, who just called a science fair project in middle school, where everybody in the family was forced to taste things that were mixed with many, many, many flavors, and I can tell you after a certain point you can't tell what's going on. So from personal experience of a middle school science fair project. Okay. Okay. So all of this says that these things that's the case. This means that odors are quote unquote sparse in odor space. Okay. So you might say, oh, good, that greatly simplifies matters. But I want to point out that this means that there is an insane number of possible natural odors that you might have to care about. Because, so the number of such odors is, roughly speaking, let's say conservatively, 10 to the 4, that's the 10,000, choose 40, which is like 10 to the 4 to the power 40, which is 10 to the 160. That is a pretty crazy number of distinct odor objects that you might choose to care about. Okay? So our goal is to try to understand how you're supposed to do this with the finite resources that you actually have to do the sensing. So you might say that, well, actually, you're misleading me because, of course, I don't care about all 10 to the 140 odors, right? I care about the smells that I care about, you know, the fruits and the animals and the things that are around me. That's what I care about. But I would argue to you that if your olfactory system doesn't have the capacity, you may care about, you know, 500 different odors, mostly because of your behavior and your niche and so on. But if you lack the capacity to sense all of these odors, if there was a region in odor space, a hole in your odor space that you could sense, I would claim that your predator will go hide in it and come and eat you because you can't smell it, right? So in the wild, I would say you at least want to have the possibility of smelling everything, just in case the new predator, you know, smells in this weird way, or you know, there's some mutation or something, it smells in this weird way, and then you can learn. Now, there's lots of learning, odor-based learning. We won't get a chance to go into this, but odor is immensely important for your memory. right? As you know, you've all had the experience of, you smell something and you get a memory back. There's a street behind the Monell Chemical Sciences Institute in Philadelphia where if I smell it, it's like I'm sitting on my school bus when I was growing up in Bombay and my feet don't touch the ground, right? I mean, it's just this sensation. And famously, there's this book by Proust called in English is The Remembrance of Things Past, where there's a scene where this guy dips a madeleine into the tea and eats it, and his whole childhood comes back to him. And then the story is about his memories, starting with his childhood. So it's a sort of famously, even in literary ways, smell evokes memory. Unfortunately, we won't have time to really talk about smell and memory in this sequence. Anyhow, so I would say that you want to smell all of these things. So our first step as theorists is to describe the space of odorants. And actually, there is a strong overlap here with a problem that people talk about in thinking about the vertebrate immune system. Because people talk about, shouldn't we be able to describe the space of pathogens? Because it's molecular sensing of 3D shapes, there should be some way of doing this. So let me describe 3 different ways you might try to do this. This chemotopic description. So this uses chemical and physical descriptors. So what do I mean by chemical and physical descriptors? You could just write down in words. This is an aldehyde. It has these additional side chains. The carbon chain is so long. This is its molecular weight, right? You can give sort of chemical and physical descriptors. It's so acidic. This is how basic it is. You can give these kinds of descriptors. And there are claims that there's a pretty good, if you take that representation, that there's a reasonable correspondence between that chemotopic description and the perceptual descriptions people give of odors. I don't actually personally believe that claim completely, to be honest. But there is a way of doing this. To me, it feels clunky because there's something we've abstracted so far away from what the molecule actually is. It sort of feels like I've sort of processed this in some way, and I'm not really sure if the processing into our terms, right, the length of carbon chain, molecular weight, really necessarily have a correspondence with the aspects of the molecule that's somehow relevant for olfaction. Maybe they do, which is why I'm putting it up here, because you should be aware of it. And maybe you will prefer it more than I do, right? A second possibility is to do a shape space description. Let me describe what this means. Imagine abstractly that I have a 3-dimensional, I have the space of molecular shapes. Obviously, this is very high dimensional, right? There's just so many degrees of freedom. This is, I don't even know how to really describe it in detail. But it's helpful to think about it that way, even here and in the immune system, because you can draw pictures like this. Suppose you have a shape space of some kind, and imagine this is a receptor, which is there in the shape space. The receptor is sort of complementary to the shape of some molecule. But really, I should draw a ball around it and say this is the region of receptor binding sensitivity. So thinking in this way is helpful because it helps you visualize why a receptor may bind to many different molecules. Because if they have shapes that are kind of within this ball, it may bind pretty well. And if it's very far away from this ball in the shape space, maybe it won't bind so well. So analytically, this kind of thing is very hard to handle, right? Because you know exactly what you do. It's conceptually helpful because you can write down equations that say, oh, there's a shape space and this abstract shape space. There's a ball in shape space that this thing does. It's cross-reactive within that ball. So I should think about all the molecules as organized in little balls in shape space, right? And then I pack the balls. And then you've sort of turned the problem into something that looks like sphere packing and things like this, which of course then maybe you can make some progress on, right? And efficient packing of spheres to describe the space, this kind of thing. So that's a very powerful thing to do and worth keeping in mind, except that we do not have an explicit description of the shape space. I should add that these days there are all these powerful techniques in machine learning So for example, as you know, or as you should know, let's see, last year, was it the year before, DeepMind published this program called AlphaFold, which does an extremely good job of predicting the three-dimensional shapes of proteins from their sequence structure, right? This famously difficult problem that all kinds of people have worked on, but this just learned to do it by having enough data. It's kind of crazy. We don't understand why it works, like everything in deep learning, kind of annoying, but it works, and that's also annoying because I hate things that work and I don't understand them. But okay, well, but there it is. That is an approach, and you might wonder whether you could employ that kind of thing to get a machine to look at lots of molecules and then give you a kind of effective description with the deep learning system of what molecular shapes are like. So that's, if no one's done it, it should be done. And one of these days I'll suggest it to a student, or maybe you'll do it before we do. Right? This is definitely a thing that's worth thinking about. So that's why I've put it up as point 2. Right? I think this was hard and very abstract, but maybe it can become concrete if you have there's a deep learning trick to somehow describe these high-dimensional spaces. Now I'm going to describe version 3, and this is a very pedestrian way of thinking about it, which is also super helpful because it's so explicit. And I'll just think in terms of concentration vectors, right? So what I mean by that is I'm going to write down C equals C1 through Cn, and N equals the number of odors, of odorants, actually, and Ci is equal to concentration of odorant I. And if I draw it this way, then it's a nice familiar representation. Here's my odorant vector, and here's where it's sort of filled up. And C is a sparse vector if I look at natural odors, because by sparse vector I mean mostly zeros, and the shaded things are, have some concentration, because I told you that of the 10,000 odorants, and then it's going to be like 40 of them turned on. So we can think about a given odor in the world as being some random set of these 40 things turned on. And as you'll see, we can use that representation very powerfully to think about the nature of odors, odor coding, efficient coding of odors, and decoding of odors. This is going to be a very helpful representation for us to think about. Okay, so this representation, of course, treats each molecule as unique, right? Which is fair. They're all different from each other. But the way in which it's not fair is the molecule you put there and the molecule you put there may be either chemically similar or similar in their shape. So it can be in this representation that things that are similar or close by in shape space get blasted far away in your representation because you don't know that they should be close by. And in any case, you can't put, you'd think, on a line, put all the proximity relations that are present in some high-dimensional space like this, right? So you're going to sort of modify the proximity relations, which may be an issue depending on how you want to think about the odors, right? So it's not faithful to the physical, chemical structure and geometric structure of things. But anyway, this is a representation, and we will use this representation often. Let me make sure. Yeah, I have 50. Do I have 15 minutes? Awesome. Great. There are more things in my notes. I'm leaving some stuff out to get through things. I may ask them to scan my notes and give them to you. Okay. So the next step, now that we have this thing, is I want to tell you about a thing that I will not treat, but which is important, which is the dynamical structure of odor space. So there's the obvious, there's one kind of dynamical structure, which involves, the long-term changes in the molecules, and really, you evolve to match the molecular space, and with the seasons, the odors change, right? We know that. If you go to, Brazil versus China, the odors change, basically geographically and with space and time, odors change. So that's definitely one kind of dynamical thing. But the thing I'm actually referring to is that in real life, if you're trying to smell, typically there's a relatively localized odor source somewhere. And then the odor comes out and forms these things called plumes. You know, it sort of moves around and there'll be little vortices like that, and it'll spread out like that. These are odor plumes. And these are highly dynamic. Odor plumes spread dynamically. And are shifted by wind. So that's by wind, I mean more like laminar flow, you know, just going, just moving things along. And chaotic currents, and chaotic air currents, by which I mean turbulent kind of currents, air currents. So in real life, this is very, very important, because one of the things you use olfaction for is to navigate. At least most animals do. We use it less, but most animals do. And actually, it turns out people are very good at navigating olfactorily. We just don't know this. There's another hilarious experiment for this Noam Sobel's lab where, let's see, what do they do? So they go out to some big field, blindfold all the grad students, and then somebody drags a turkey on the ground and takes it all the way to some other end of the field. And they tell the grad students, find the turkey, right? And then all of them get down on their knees and go, and they're very good at it. So I think it's partly we're less good at olfactory navigation because we don't pay attention to these cues. And also there's stuff like if you get down on the ground, close to the ground, you can smell more molecules because someone is sitting down there, so it's easier. And actually humans, you know, have remarkable abilities. There's a lady in England who was reported not so long ago, who her husband was dying of Parkinson's paralysis, and she goes to the hospital where there were lots of Parkinson's patients, or supposed to be. Sorry, no, that wasn't the way it went. She felt her husband smelled sort of sweet in a particular way, right? There was a smell. And she kept telling the doctor, and he said, what are you talking about? Then finally, on a hunch, he took her to the hospital. And she walked past people and said, well, this person smells sweet. That person smells sweet. And the ones she identified, most of them had Parkinson's. And of the ones who didn't, a substantial number developed it afterwards. So you can smell this. And you know, you can smell COVID. Dogs, they've trained in some places dogs to smell COVID and things like this. So actually, we have remarkable olfactory abilities. We just don't focus on it, right? So, but we could train it. Okay. So anyhow, you can navigate, or many animals navigate. And there's a professor from Berkeley whose name I'm temporarily forgetting, who claims that olfaction is primarily for navigation and not primarily for identifying things based on their smell. Okay? So because of that, these kinds of dynamical things are very important, and all of the circuits have many dynamical features. They've got oscillations, they've got transients, you know, there's lots and lots of dynamical things, which I don't have time to talk about, right? Some of that I'll allude to it a little bit. A lot of those things you really have to talk about the dynamics of circuits, right? And the details of how, you know, the things I said, I was going to defer to Professor Capelli to discuss. You know, you make the network has some excitation, some inhibition, there are timescales in various places, it reverberates, you know, things like this. These things matter for this kind of thing, right? So we don't have time for it. I will leave it to Professor Capelli to discuss how you model such things. So that's my caveat. So, and I'm going to put down here, we will instead, so this is important for navigation. So we will instead focus on static combinatorial coding properties, and basically trying to answer the question, the categorical question, what did you smell? Not where did it come from. So this is really a distinction between what questions and where questions. Okay? There was a question there?
I would just ask if you have any clue about which molecule she was smelling to find parts.
I'm the same as you, as in I've just been reading the press and I haven't actually followed the details. During the COVID times, I was still finishing some of my papers in olfaction, so I found it really interesting that some COVID patients lose their sense of smell. So I attended an entire conference as an audience member, which was talking about this phenomenon. And I came away still not knowing exactly what was going on. So I think there's a lot that's unknown. In those cases, in the seminars that I went to, there was a lot of discussion about how bitter compounds and stuff like that are often associated with bacteria because they secrete these things. So one of the reasons why we have the ability to taste and smell bitter things is because it helps detect infections. At least that was the claim. But when you detect these things, sometimes what happens is that you emit inflammatory substances, partly because you think that you have a bacterial infection or something and you want to kill them. And so you get inflamed. And sometimes this can have the effect of damaging your own system. Overinflammation can damage you. And so that at least this is 2 years ago, so I'm sure the science has moved way forward from then. But this is just my snapshot from 2 years ago. So I think a lot's not known about this kind of thing. I mean, there are other also sort of funnier studies. Like I remember one goofy study from somewhere in California where they went to some college and they took a bunch of men, a bunch of women, had them wear T-shirts and work out in the gym, and then they passed the T-shirts to the other gender and said, "Sniff it and tell me which of these people you would be happy to go out with," right? And people make their selections. And then there was a claim made that there was some sort of compatibility between the MHC complex and the immune system. You read these things and you're like, maybe, you just maybe, it's not definitive. But there's lots of anecdotal stories like this. So I'm just trying to say, you can make lots of anecdotes, but this needs to be put on a more firm footing as to whether it's actually true that we're able to use our olfactory systems to sense these kinds of things about the health of ourselves and other individuals and things like that. If we can, it's a very useful thing, and this is something we need to know about. Right? Yeah. Okay, so I have like 5 minutes now, and I'm going to use it like I usually do at the end of a class to set up what I'm going to do for the next class, right? So the thing I want to do here is talk about the circuits for olfactory processing, because the entire next lecture is going to be based on rapidly going through sections of this. By the way, it will go faster if I do things on slides, but you won't be able to take notes. So what's your feeling about that? You prefer the blackboard? Okay, in that case, I'll just cover less, but then what I'll try to do is I'll try to hand out notes on the stuff I don't cover. Okay? So I want to tell you about the conserved olfactory circuit that's conserved from flies to humans. So we've already talked about odor space. There's lots of odors, they float around, odorants, they float around. And these are natural odors. So that's the front end. And I won't say more about that now. Now, if you now go look inside the I'm going to talk as if I'm talking about vertebrates, and then I'll tell you the insect version of it. For vertebrates, there's a thing called the olfactory epithelium, right? It's a membrane in which you've got olfactory receptor neurons embedded. So I'm going to draw them. And so these are ORNs, olfactory receptor neurons. In flies, these occur they're called olfactory sensory neurons, and there isn't an epithelium, right? There's another structure in which these things are stored, right? And they're called olfactory sensory neurons. And it turns out that there are different kinds. So you'll find that a neuron will express one receptor, right? And it's different receptors. And it's a very interesting problem in developmental biology to understand how the neuron manages to select this receptor as opposed to that receptor and then prevent two from being expressed at the same time. Right? So cool problem in development, okay, and cell biology. So what they do, and it turns out each receptor has a separate gene. In fact, the olfactory gene olfactory receptor gene family is typically the largest gene family in any animal, right? By far. Okay. So there are about 50 types in Drosophila. And there are 50 types of neurons, 50 types of sensors, if you like. There are about 300 in human and about 1,000 in mouse. And just to emphasize, so you know, we're much bigger than fly. That's part of the reason why we have more. But notice a mouse is much smaller than us and it has more. And just to be clear, that you can go all the way to the African elephant, which is the biggest land animal, and it has 2,000. So one of the things that I find very interesting and puzzling about the system is from the smallest to the largest creatures, and I don't mean worms, because worms will have about 10 of these types, right? So, well, but even that's like this. I would say it's all about a few hundred, like 300, 400, give or take a factor of 6, right? And the African elephant is a billion-fold heavier than Drosophila. All animals have an operating point, I would say, of a few hundred receptor types, and it seems to be sufficient for animal behavior, right? So I'd like to know why that's the operating point. Why isn't it I need 5,000 receptor types, or I need only 10, given that I can have this sort of combinatorial matching of many molecules to many receptors. So the next stage is called in vertebrates the olfactory bulb. So this is the bulb. And I'm not going to write down, maybe tomorrow I'll write down the insect lingo. For insects, it's called the antennal lobe. And you'll find that all receptors of a given type, all neurons with receptors of a given type, project to a structure called a glomerulus here, which sort of aggregates the signal. And we're going to use this in our modeling. So, you know, all the white receptors are going to this glomerulus, all the black receptors are going to this glomerulus, and so on and so forth. They collect them. And there's an interesting structure with a circuit and, you know, the interconnections that is not that well understood exactly what it does. Then an interesting thing happens. These things have kind of project into, still within the olfactory bulb, into things called mitral cells. And there's some cross projection, so it's not just one-to-one. There's a layer here in which there are, in this circuit. And notice, it looks like one of these classic neural network models with some sort of feed-forward circuit and so on. There's a layer here with so-called mitral cells. And mitral cells are excitatory cells. So for those who haven't done neuroscience, there are two basic kinds of neurons. There are excitatory neurons and inhibitory neurons. Excitatory neurons have the property that when they fire, they cause the downstream neuron at the next synapse to fire. And inhibitory neurons have the property that when they fire, they make the downstream neuron one step down, the next synapse down, to be inhibited, to stop it firing. And there's a thing called Dale's law that says never the two shall meet, right? I mean, that you either have excitatory or inhibitory synapses. I don't think that's 100% true, but it's kind of basically almost 100% true, right? In general, it's one or the other. So these are excitatory cells. And so they are going to be responsible for doing whatever processing they need to do and send this further down. And also here, there are so-called granule cells. And the granule cells are inhibitory. And these things take inputs from mitral cells and then inhibit other mitral cells, and they inhibit granule cells, and they inhibit themselves. And so it's a sort of complicated circuit. So this is an excitatory inhibitory network with very interesting properties. In fact, my last PhD student, he was an MD PhD student, the core of his PhD thesis was actually building a detailed mechanistic model of the kind that I usually really do not like in this system. So he went through all the anatomy and worked through a very detailed sort of model of how everything connected. Because, you know, in these kinds of networks, the range of connectivity matters, right? If one of these inhibitory cells can inhibit the entire structure, it's going to have a very different dynamical effect than if it only inhibits in the local neighborhood, right? So the connectivity and everything matters. And there's a lot of, in the way, if you do this kind of work, you'll find that there's enormous amounts of anatomy and physiology done, but it's all in separate papers, right? Everybody's doing different things. So you just have to spend, you know, a decent amount of time reading all the papers and collecting it. So basically what he did was he built a mechanistic model putting all of this thing together and justifying that A, it reproduced what you see physiologically, and B, it made some new predictions. I'll tell you about that later. Anyway, so there's this excitatory inhibitory network that reorganizes the information that's come in and mixes it and subtracts it and normalizes it. I'll say more about that later in some way. So we won't do a detailed model of this network, but I will describe at one point an effective model that's roughly capturing a part of the net effect. Then comes the next stage, which is for vertebrates. This is the olfactory cortex, sometimes called the piriform cortex. And in flies, this would be called the mushroom body because it looks like a mushroom, right? Now here's something completely crazy happens. So you take a given mitral cell, and in every other sensory system, what happens is that this output cell in the retina will project in a structured way there and there and there, or it's all very structured, highly organized when it projects the cortex. Here, it appears to be statistically random. So this thing will make projections to some random collection of cortical neurons, and this will do it randomly. And I mean, it looks statistically random. This is done through tracing studies. What you do is you inject a virus that will go back down in a given mitral cell, that will go through the axon of the neuron and basically jump into the postsynaptic neuron, and there it inserts itself into the genome, and causes the expression of a fluorescent protein, and thereby you can see where it projected to. So you do these studies, and as far as we can tell, it's statistically random, both in vertebrates and in flies. So this is clearly a theme here that's developing. This looks kind of random, the binding of molecules to the receptors. This doesn't seem to be any organization. As you'll see, if you write down the sensing matrix, it just looks like this kind of splotches everywhere. It's kind of random, right? And then this looks kind of random. So one of the questions I would have looking at that is, wait a minute, this looks so different from every other sensory system I know. It looks all kind of random. What's going on? And maybe there's a reason why it's random, and I'll try to argue to you that randomness in the circuit of this kind, structural randomness, helps with the efficiency of the coding and the effectiveness of the decoding of the odor. Because cortex is where, so this looks like random projections. And the reason why I'm saying that, talking about decoding, is what happens is once you get the information in cortex, other regions of the brain take input from this, and they're charged with making decisions. For example, you may need to learn that all of these odors are appetitive, namely they're good and you can go towards them and maybe eat them, and these things should be aversive, namely you should go away from them. Here I'm talking in the language of behavioral studies where things are appetitive or aversive, and so those are learned things, and even flies can learn them. You may think they're innate, but you can get them to learn that the banana odor is terrible for them because they'll die or get sick. They can learn many things. So these are learned, right? So that's learned based on the representation here. So we're going to look at the circuit, and we're going to sort of pull it apart and think about what it means tomorrow. That's going to be the whole day tomorrow. Okay? Sorry I ran over.